So in the last video, I talked about brain tissue slices, why they're important to neuroscientists, why they're popular to work with, and what scientists can learn from working with them. In this video, I want to focus on one challenge when it comes to working with these tissue slices, and that is keeping the tissue alive for several hours. So since we're just talking about a piece of tissue, there is no heart, there is no lungs, there is no blood circulatory system, which usually provides the tissue with all the oxygen and all the nutrients it needs to stay alive. It's just a piece of tissue in that holding chamber. So how do we keep it alive for several hours in order to work with it? So neuroscientists perfuse the tissue with something that's called artificial cerebro-spinal fluid, or brain juice. It's basically a solution that imitates the extracellular solution in the brain. It can be made relatively easily. It has mostly sodium chloride in it and several other salts. It is also bubbled with oxygen to oxygenate the solution and then the tissue. And in many cases, it also has some form of sugar, such as glucose, added, so the tissue can stay metabolically alive. And then the chamber where the brain section is located has two tubes connected to it. One is the inflow, one is the outflow. And the inflow tube has a constant inflow of this brain juice into the chamber to superfuse the tissue, basically run past the tissue and oxygenate it, feed it, and so forth. And then the outflow tube collects the spent solution and puts it into a trash can. And so with this technique, doing the experiment that lasts for 4, 6, 8 hours, a scientist may go through several liters of brain juice over the course of a given day. That's usually not a problem because that brain juice is really cheap to make, doesn't contain anything terribly expensive. And so even using liters of it is not a big deal financially. However, in many cases, a scientist may want to add some pharmacological agent to that solution, and that's where things can start to get really, really expensive. These pharmacological agents are used to block particular aspects of the cell physiology, for example, ion channels, synaptic blockers, the connections between neurons, and so forth. And many of these agents are, for example, spider toxins or scorpion toxins, pufferfish toxins, or they're artificially made with great effort. And you can imagine they can be really, really expensive. In fact, by weight, they can be multiple times more expensive than, let's say, gold or diamonds even. So whenever a scientist works with one of these pharmacological agents, the goal is, in order to keep the experiment affordable, is to use as little of this brain juice as they possibly can. And so what are methods to reduce the consumption of brain juice over the course of an experiment? One could, for example, reduce the flow rate. A typical flow rate of brain juice across the tissue is, let's say, 8 milliliters per minute. This could be reduced to 6, 4, 2, 1 milliliter. Any reduction that is too much, however, would risk that the oxygen escapes from the brain juice and would not keep the tissue alive. So the reduction of flow can only go so far. The next method is, how about not throwing away the spent solution, but reusing it? So scientists can connect the outflow of that chamber back to the holding tank and make the outflow recycle into the inflow. So once that spent solution is back in the holding chamber, it can be reoxygenated and then used a second, third, and fourth time. And that can, of course, greatly reduce the amount of solution that is used on a given day. You can only, however, go so far. Like using an inflow tube and an outflow tube and a holding container still requires a minimum amount of solution. And in our experience, it's maybe 7 to 10 milliliters. So that means you still need at least 7 to 10 milliliters of brain juice for a given experiment on a given day to get a flow going and get a circulation of brain juice across the tissue that doesn't break down and that functions well. So how can we reduce these even further? Because even 10 milliliters can get quite expensive with certain agents. So here's where PopNeuron's ultra-low volume tissue chamber comes in, which reduces the amount of brain juice for an experiment to about 1.5 to 2 milliliters. And in the next video, I will talk about how that is being accomplished.